Good day to all of us. I am Rafa Jane Obanan, joined by my group mates from Group 3, Manlangit, Okada, Quesada, Salud, Suminod, and Tado. Today, we will delve into the fascinating topics of oxidative phosphorylation and electron transport. So, sit back, relax, and lend us your ears as we explore these crucial biochemical processes. But first, let me ask you, why do we need to study and understand oxidative phosphorylation and electron transport in biochemistry? Well, the answer is oxidative phosphorylation and electron transport are fundamental processes in biochemistry that drive cellular energy production. Understanding these processes is essential for comprehending the biochemical basis of energy metabolism. These processes involve a series of biochemical reactions that converts energy stored in molecules like NADH and FADH2 into ATP, the primary energy currency of cells. Knowledge of these reactions is crucial in biochemistry to understand how cells generate and utilize energy. Now, what is oxidative phosphorylation? Well, oxidative phosphorylation is the last stage in the process of cellular respiration following glycolysis and the citric acid cycle. So what is cellular respiration? Well, cellular respiration, it is a process through which cells generate energy by converting nutrients into ATP, and ATP is the molecule that provides energy for various cellular activities. And also, it involves electron carriers like NADH and FADH2 transferring electrons through series of protein complexes in the mitochondria's inner membrane to oxygen. And this process occurs within the mitochondria of eukaryotic cells, specifically in the inner mitochondrial membrane. And the main purpose of oxidative phosphorylation is to harness the energy from electron transfer to generate adenosine triphosphate or ATP, which is the cell's primary energy currency. So as you can see on the screen, no, that is the structure of oxidative phosphorylation. No, it is a series of compartment and complexes within the mitochondria, which is an organelle found in the eukaryotic cell, and it involved first the intermembrane space. So this is the space between the inner and the outer mitochondrial membrane. So it is not directly involved in the production of ATP, but plays a role in the overall structure of the mitochondria. Second is the inner mitochondrial membrane. So this is the innermost membrane of the mitochondria where the electron transport chain and ATP synthesis are embedded. So it is highly folded, increasing its surface area and providing ample space for these complexes. Third is kanang H plus, no, gan kay H plus, that's protons, no, are pumped from the mitochondrial matrix. No, the innermost compartment of the mitochondria to the intermembrane space during the electron transport chain process. So this creates a proton gradient with a higher concentration of protons on the intermembrane space side. And fourth is, makita ninyo na ay ADP plus then PI. So that stands for adenosine diphosphate then inorganic phosphate. So these are the precursors of ATP, which is the primary energy currency of cells. ADP and PI are converted to ATP through the action of ATP synthesis, which uses the potential energy stored in the proton gradient. And lastly, we have the ATP, which is adenosine triphosphate. So this is the final product of oxidative phosphorylation, which is highly energized and can be used by the cell for various metabolic processes. In summary, The structure of oxidative phosphorylation involves the movement of protons across the inner mitochondrial membrane, creating a proton gradient. This gradient is then used by ATP synthesis to convert ADP and PI into ATP, which is again the primary energy currency of cells. And on our next slide, we have the two stages no, of oxidative phosphorylation, which is the chemiosmosis and electron transfer chain. So chemiosmosis, which uses the potential energy stored in the proton gradient to convert ADP and PI into ATP. And then the electron transfer chain, which pumps protons and create a proton gradient. So these two no stages work together to generate a large amount of ATP from a relatively small amount of energy stored in the oxidized nutrients. In conclusion, oxidative phosphorylation is a pivotal or important process in cellular respiration that efficiently converts the energy derived from electron transport into ATP, serving as the primary energy source for cellular activities and highlighting its critical role in biochemistry.
We'll explain briefly the steps of oxidative phosphorylation. I will be breaking down each stage into simpler terms. Now, the first step is electron transport chain initiation. The process of oxidative phosphorylation begins with the initiation of the electron transport chain. This is like a starting point along the journey where electrons are passed from one molecule to another. The electrons are first transferred to the protein complex 1 in the inner mitochondrial membrane. This kickstarts the flow of electrons through the chain. The second step is electron transfer through protein complexes. Here, as the electron move the electron transport chain, they pass through a series of protein complexes such as complexes 1, 2, 3, and 4. This is like a relay race. Each protein complex plays a specific role in accepting, transferring, and passing on the electron to the next complex, creating a flow of energy. The third step is proton pumping and gradient formation. During the electron transport process, proton with H plus ions are pumped across the inner mitochondrial membrane from the mitochondrial matrix into the intermembrane space. This creates a concentration gradient with more protons on one side of the membrane than the other, setting the stage for the next crucial step. The fourth step is ATP synthesis via chemiosmosis. The buildup of protons on the one side of the membrane creates a potential energy difference similar to a charged battery waiting to be used. This potential energy is harnessed through a process called chemiosmosis, where the flow of proton back across the membrane drives the synthesis of ATP by the ATP synthase enzyme. The last step, which is the ATP production. The final step of oxidative phosphorylation results in the production of ATP. The energy currency of the cell is ATP. ATP is like a rechargeable battery that powers various cellular processes. The ATP generated through oxidative phosphorylation is essential for activities like muscle contraction, cell division, and other energy demanding function in the body. By understanding these intricate steps of oxidative phosphorylation, one can appreciate the remarkable process through which cells efficiently convert energy from food into ATP, ensuring the continuous supply of energy needed for cellular activities and overall functioning of the body.
understand what electron transport is. So, electron transport refers to the process of transferring of electrons along the series of protein complexes and molecules, leading to the generation of ATP through oxidative phosphorylation, which serves as a vital mechanism for energy production in cells. Then, electron transport occurs in the inner mitochondrial membrane in eukaryotic cells and the plasma membrane in prokaryotic cells. So, why is it known that the electron transport in eukaryotic cells occurs in mitochondria while prokaryotic cells occurs in the plasma membrane? So, in eukaryotic cells, electron transport occurs on the inner mitochondrial membrane due to the presence of specialized structures within mitochondria called cristae. So these cristae provide a large surface area for the attachment of the protein complexes involved in electron transport. Then the inner mitochondrial membrane is highly folded, creating numerous compartments where the electron transport chain proteins can reside and functions efficiently. These organizations allow for the effective transport of electrons, and the establishment of a proton gradient is essential for ATP production. On the other hand, prokaryotic cells is walay mitochondria. That's why electron transport takes place on the plasma membrane. The plasma membrane of prokaryotic cells serves as the similar function to the inner mitochondrial membrane in eukaryotic cells, providing a platform for the electron transport chain proteins to function and generate ATP. So as what you can see in the picture, it is a process. So the purpose of electron transport is to create a flow of electrons that drives pumping of protons across a membrane, establishing a proton gradient. Then this gradient is then utilized by ATP synthase to catalyze the production of ATP, which serves as the primary energy currency within cells. Then by efficiently transferring electrons along the electron transport chain, cells can extract energy from nutrients and convert it into usable form for cellular activities. So this process is essential for cellular respiration and plays a vital role in the overall energy metabolism of organisms. So to sum up, electron transport and oxidative phosphorylation are interconnected processes that enable cells to harvest energy and convert it into ATP, facilitating various cellular function, which is essential for life.
And now, let's talk about the electron carriers. So what are electron carriers? Electron carriers are small organic molecules that accept electrons from an electron donor, as well as donate them to electron deficient molecules. Thus, working as an electron shuttle system, electrons shuttle from one molecule to another via electron carriers. So we have here the first electron carrier, which is the NADH, that stands for the nicotinamide adenine dinucleotide. So this electron carries electrons gained from the breakdown of glucose and donates them to the chain of enzymes in mitochondria that are involved in producing ATP or electron transport chain. Also, this oxidizes NADH back to NAD+. So this term is actually used to describe reduced and oxidized forms of the same molecule. The NAD+ is the oxidized form, that is a state in which it loses an electron, while NADH is a reduced form of the molecule, which means that it gains the electron lost by NAD+. Now, next we have the FADH+. So this electron carrier is responsible for transporting electrons generated in major pathways like glycolysis and Krebs cycle. to the electron transport chain. It also has a role in cellular respiration. It acts as an electron carrier and temporarily restores energy during cellular respiration. So this the FADH plus contains 2 electrons, and this is a process of reduction which stores the energy in high electron states in the FADH2. FAD can carry 2 electrons. FAD also functions as an electron carrier in various processes such as cellular respiration. So both the NADH and the FADH2, its role is to donate electrons to the electron transport chain. They also both donate electrons by providing an hydrogen molecule to the oxygen molecule to create water during the electron transport chain.
So we have here the 4 complexes. First, we have the complex 1, which is the NADH dehydrogenase. Complex 2, the succinate dehydrogenase. Complex 3, the cytochrome BC1 complex. Complex 4, the cytochrome C oxidase. And the complex 1, or the NADH dehydrogenase, is the first and largest protein complex in the electron transport chain. It receives electrons from NADH and passes them to ubiquinone. Ubiquinone is a nutrient that the body needs in small amounts to function and stay healthy. Complex 1 facilitates the transfer of electrons from NADH to coenzymes through FMN, or the flavin mononucleotide prosthetic groups, and the FES, or the iron sulfur clusters. So seamlessly and transports proteins across the inner mitochondrial membrane, generating the electrochemical gradient necessary for ATP synthase. So next, we have also the complex 2, or the succinate dehydrogenase. So it is involved in both the citric acid cycle and the electron transport chain, and it is also accept electrons from succinate and transfers them to ubiquinone. So in complex 2, function involves the oxidation of succinate to fumarate utilizing FAD prosthetic group and coenzyme as intermediate. So it is distinct from complex 1 in that it does not aid in generating the proton motive force necessary for ATP production.
So for the next slide, we're going to tackle the complex 3, or cytochrome BC1 complex. So cytochrome BC1 complex is responsible for transferring electrons from coenzyme Q to cytochrome C. So it plays crucial role in transferring between first and second halves of electron transport chain. So cytochrome to iron cluster and iron cluster to cytochrome A. The primary function of the complex 3 is to transfer electrons from ubiquinol to cytochrome C. This process, coupled with the translocation of protons across inner the mitochondrial membrane, which contributes to the protein gradient used by ATP synthase to produce ATP. By facilitating the transfer of electrons and pumping of protons, the cytochrome BC1 complex plays a crucial role in the generation of ATP through oxidative phosphorylation. This process is essential for the energy metabolism and aerobic organisms. For the next slide, we have the complex 4, or the cytochrome C oxidase. So it is a final enzyme complex in the electron transport. It receives electron from cytochrome C and transfer them to oxygen, the final electron acceptor, to form a water. Cytochrome C to cytochrome A and cytochrome A to oxygen. Complex 4 is essential for cellular respiration, energy production, and maintaining cellular health. Its proper function ensures efficient ATP synthesis and protection cells from oxidative damages, making it critical component of mitochondrial biology and a significant focus in medical research.
There are three inhibitors of ATP synthesis. So the first inhibitors is the electron transport. The second is inhibitors of oxidative phosphorylation. The third is the inhibitors or uncouplers of oxidative phosphorylation. So for the first inhibitors, which is the electron transport, NADH or the nicotinamide adenine dinucleotide, FMN, which is the complex one, and coenzyme Q, it inhibits the electron to nicotinamide or the NADH. And this inhibitor says that chlorphomazine, rotenone, anobarbital, and pyridine, which is antibiotic. Then the transport of electron from complex 2 to coenzyme Q, it is inhibited by carboxyl. Inhibits through of electron FAD to coenzyme Q. Malonate is a competitive inhibitor of succinate. Then the flow of electrons from complex 3 to cytochrome C is inhibited by BEN, which is baldimetoprol, antihistamine A, and naftoquinone. The flow of electrons from complex 4 finally to oxygen. It is inhibited by carbon monoxide, cyanide, oxide, and hydrogen sulfate. So this is the inhibitors of electron transport. So next is inhibitors of oxidative phosphorylation. This oligomycin inhibits F1 ATPase by blocking proton flow through FO complex. Orovertin inhibits F1 and atractyloside inhibits ATP exported by ADP translocase, and those are the inhibitors of oxidative phosphorylation. So the third is the uncouplers of oxidative phosphorylation. Dissociate oxidation from phosphorylation, and these uncouplers are toxic lipophilic compound which readily diffuse through mitochondrial membrane, and they make the mitochondrial membrane permeable to protons, and because of this, it prevents formation of proton gradient. Those energy produced by transport of electrons is released as heat, which is not ATP. Next is the diseases related to oxidative phosphorylation and electron transport. The first disease is cancer. As we all know, cancer is a disease in which some of the body cells grow uncontrollably and spread to the other parts of the body. Cancer can start almost anywhere in the human body, which is made up of trillions of cells. And the second disease is the neurodegenerative diseases or disorders. Example of this are Alzheimer's disease, another memory disorder, ataxia, Parkinson's disease. Yes, those are the examples of neurodegenerative disorders.